Hi guys. I'm glad you can be here for my AP Biology presentation. So it's 7, so I'll go ahead and get started. So today I'll be going over AP Biology. First we'll start with the course content. So just all the units that we need to learn, and like just an overview, pretty general. And then we'll be going through a detailed dive of the free response. So just like specific questions and how it's going to work. So there are 8 units in AP Biology. Here are all the 8. The percentages of how much they are tested on the exam are on the side, just for reference. So we'll go ahead and start with Unit 1, which is Chemistry of Life. So this unit is generally just all biochemistry. So the first big thing is water. If you think about it, water is the biggest thing of life. For example, if you're going out into space, you'll want to be looking for water on planets to determine if there's life or not. So water is basically the big bringer of life. So you learn about water, its properties, the special properties that it has, like for example, the surface tension it can hold, or the way that when it freezes, it becomes less dense than if it was not frozen, if it was in liquid form. So just general information about water, which is a big part. And then there's the introduction to biological macromolecules. So the 4 big biological molecules that we have in our bodies, which are carbohydrates, proteins, lipids, and nucleic acids. So these 4 macromolecules are all broken up, have smaller parts called just monomers. So the monomer of carbohydrates is a monosaccharide. For proteins, it's an amino acid. For lipids, like a triglyceride, it'd be 3 fatty acids and a glycerol. And for nucleic acids, it's nucleotides. So then you learn about the properties of biological macromolecules, which is basically the properties of the monomers and the types of bonds that connect the monomers in these biological macromolecules. So basically you learn about the structure and how they form. For example, proteins have different levels of structure, and then like nucleic acids are built differently, and carbohydrates are all sugar. Okay. Then we go on to structure and function of biological macromolecules. So the one big thing in biology is that structure defines function. So the structure of your, whether it be a molecule, a cell, or anything, determines how this thing will function. So for example, the picture below, you can see it's starch, glycogen, and cellulose. So these are all different types of carbohydrates or different types of sugars, but they're all for different purposes. So the way it's structured, for example, cellulose on the very right is structured very neatly and compactly, which allows it to be what holds the plant's shape in the plant wall. So cellulose is what makes plant walls. It also is what makes it indigestible for humans, while starch and glycogen are both energy stores in different ways and that we can digest. So structure defines function. Then there's a deeper dive into nucleic acids. So basically you get a general structure and then some other stuff that you need to know. One thing is that just for future reference, I might talk about this a little in future slides, but in nucleic acids, there's a phosphate group, a base, which is either thymine, cytosine, adenine, or guanine, and then a 5-carbon sugar. So the carbons are counted 1, 2, 3, 4, 5. So later when we talk about DNA or something and you hear 3' to 5', it just means the 3rd carbon to the 5th carbon, or 5' to 3', which is 5th to 3rd. So it's just the way it's organized and how it flows, basically. Continuing on, so Unit 1 is basically just basics. It helps you learn stuff that you will need for all the future units. Okay, so the next unit is unit 2, which is cell structure and function. So here you have the cell structure. So you learn about what makes up a cell, all the components of it, and how these components work together. Then you have cell structure and function, which is like what the structures are and how it works. Then you learn about cell size. The big thing in cell size is that you need to know that surface area to volume ratio needs to be big, which means the smaller the cell, the better, because so surface area is the outside of the cell, while the volume is the stuff it contains. So in this case, having a larger surface area allows you to import more things into the cell, while having smaller volume allows the cell to be able to transport stuff inside and outside faster because there's less distance to travel. This means that in the end, having a smaller cell, having a smaller thing makes the surface area to volume ratio higher, and therefore why cells are small. Okay, and then we move on to cell compartmentalization, which is just organelles basically. So like the different organelles, how they work, what they do, pretty important. There are a couple big ones that are like the most important, which are ribosomes, mitochondria, and that's it. It's pretty important to know everything else, but those are just the big ones, and the nucleus, of course. But those are just the big ones that you need to know. Then there's the origins of cell compartmentalization. There's this thing. Okay, so on Earth, before like humans occurred, before animals, before like all of that, there was only bacteria, and the origins of cell compartmentalization were basically that one bacteria, ate another one, and then, but the big bacteria that ate the smaller bacteria didn't digest the smaller bacteria, and instead the smaller bacteria started working inside the bigger one for an exchange. So they started forming like there was a relationship in there. So the big bacteria provided shelter and was able to get food and energy for the smaller bacteria, while the smaller bacteria was able to do stuff like, for example, convert energy for the larger one. So that's how the origins, started, mutualism, for somebody who said. So they started working for each other to make the cell more effective and more efficient. This is in the end how bacteria formed into eukaryotes, which is the cells that we're made up of. Okay. Anyways, then we move on to the plasma membrane. I spent a long time learning about this, like our class spent a long time, because it's quite an important part and plays into a lot of stuff. So the plasma membrane is made up of phospholipids, which have a hydrophilic head and hydrophobic tails. So the head is attracted to water while the tails are repelled by water, which is why they form this formation. The tails group together while the heads go on the outside. This proportion is just to show you how the different portions, but in general, the hydrophobic tail is much longer than the hydrophilic head. Okay. So plasma membranes are pretty important. They separate everything. They separate the cell from the outside environment, and the membrane permeability, which is basically plasma membranes are selectively permeable. Only certain things can go through, and it keeps out other things, which is why there are things like these weird channel things. They're called protein channels or membrane proteins that allow things to go in and out selectively. Okay. Then there's continuing on to the membrane portion is that there's membrane transport. So like I said, because the membrane is selectively permeable, only some things can go in and out. It has to keep most things out. And so this is where membrane transport comes into play. So it is able to help move from one place to another if the if the thing cannot go through by itself. For example, those channels are a form of membrane transport. Then there's facilitated diffusion. So diffusion, so all of these things are to do with like diffusion, osmosis, and like moving from one side of the membrane to another, stuff like that. I'm not really going to go into detail because I don't think we'll have enough time for that. But anyways, so in this unit, for our class, we did a lab which tested the osmosis in potatoes. So if you take AP Biology, you might not do this specific lab. You might do other ones. But for us, we did one that determined how water moved through a potato when put in different concentrations of sugar water, and then to determine like the molarity of the potatoes and the water potential. So the next unit is cellular energetics. So big thing, enzymes. So enzymes are a type of protein that help catalyze a reaction, so help speed up a reaction by lowering the activation energy. So you'll learn about the things in detail if you do go into AP Biology. Yeah, that's enzymes. They're pretty big. They're pretty important in a lot of other things and like experiments and stuff. So it's good to know how they work and what they do. So energy. Energy, everybody needs it. All living things need energy to survive and do metabolic processes. So, but depending on what the thing is, what the living thing is, depends on it. The way it gets its energy depends on what this thing is. So if you start with plants, for example, plants get their energy from the sun. Through photosynthesis, they're able to produce glucose, which is sugar, for themselves to be able to survive. So here's a simple equation, which is usually what you would probably learn in like middle school. But in AP Biology, you'll delve more into like how the specifics of how it works and all the processes like the Calvin cycle and all these energy transfers and just a lot more details. Quick note though is that on the AP Biology exam, you won't be required to remember any specifics like, oh, the Calvin cycle needs this thing right here to like function or something. You don't need to know any specifics, but you do need to know general of how it works and like the steps. And that's just for everything. So you won't ever need to know anything specific like names of specific compounds or any of that. Just know the general idea and that'll be fine. But that doesn't mean you shouldn't learn it. It's just not going to be tested on the biology exam. Okay, then there's cellular respiration. So I guess as opposed to photosynthesis where plants do it, cellular respiration is what animals and like humans do to get our energy. So this process starts off with glycolysis, which is breaking out glucose down into pyruvate. This is why we need to eat, is because we need to be able to get this glucose from somewhere, which is our food source. So this glycolysis step happens out in the cytosol, so out in the cytoplasm, and not in the mitochondria, which is where everything else happens. So then there's pyruvate oxidation, which is just oxidizing the pyruvate, and then it moves into the mitochondria where the citric acid cycle or Krebs cycle happens. You'll learn the details about what happens in the Krebs cycle, but again, that's probably not tested on the exam. Then after the Krebs cycle, there's electron transport and then ATP synthase. So cellular respiration provides ATP, which is the standard like unit of energy for humans, actually and just in general. But ATP stands for adenosine triphosphate, and the way it provides energy is that the triphosphate means there are 3 phosphates attached. So one of them can be broken off and then the energy will release and it'll become ADP, so adenosine diphosphate because there are only 2. And then if another phosphate gets added back on, it becomes ATP again. So it stores energy. Then after cellular respiration, we learn about fitness, which is just how well fit an organism is to its environment. Yeah, we'll go into deeper about that in another unit. Yeah. Then we go to cell communication and the cell cycle. So cell communication, the two big forms of communication in the human body are the nervous system and the endocrine system. So I went into more detail about these two systems in my psychology presentation, but the reason they're mentioned in biology and psychology are different. So yeah, that's just a note. So all you need to know really for biology is that the nervous system works by functioning by transmitting neurons through neurotransmitters, while the endocrine system transmits hormones through the blood. With cell communication comes feedback, which are loops. So basically, a stimulus. So there are two big types of feedback. There's negative feedback and positive feedback. So this doesn't have anything to do with like if they're good or bad. It just means that for negative feedback, a stimulus is sent. A stimulus happens, which causes a signal to be sent. A response happens that decreases the stimulus. For example, in blood sugar, for example, if you eat something and your blood sugar rises, a signal is sent through your body, and then the response is just like insulin into your blood, and then release insulin into your blood to decrease this high blood sugar level. So lower your blood sugar. Negative feedback, these loops are the most commonly found. So positive feedbacks are sometimes, for example, which is basically there's a stimulus, a signal, and then the response increases the stimulus more. This happens in like if you have childbirth, so like a stimulus, like the child is ready to go out, so the response is that like there are contractions, and in the end the stimulus increases until like the contractions keep on happening until the baby is out. Okay, so from the feedback thing, there was this signal sending portion. So this is where the signal transduction comes into play. So signal transduction has three big portions: reception, transduction, and response. So reception is this part right here. So this part is where like the signaling molecule is received by usually a membrane protein, and then that's just the reception part. Then it goes on to transduction, which is basically a cascade of events that happen until it hits the response, which is whatever the signal was meant to go do. So if there are any small changes in this pathway, the response will most likely not happen. For example, if this receptor suddenly had some sort of change in the structure, like some small change, then it might not be able to have the signaling molecule fit into it. So this whole chain of events wouldn't happen and the response wouldn't be induced. This is how function follows structure. Like the receptor, the structure of it was wrong, so it wasn't able to function the way it was supposed to. Okay, then the cell cycle. So if you took biology, like introductory, any sort of like 7th grade middle school biology, you got to remember like mitosis. So this cell cycle refers to how cells are able to replicate. So interphase, the cell cycle is split into interphase and mitosis. So interphase is just like everything that's not mitosis, and mitosis is the splitting portion where the cell is actually doing, like actually physically doing things inside to split. Okay, so interphase contains 3 different portions: G1, S, and G2. So G1 is gap 1, which is where the cell is just growing larger and larger, getting ready to split. Synthesis is where DNA is replicated, and then G2 is another phase of growing until finally you hit mitosis. So mitosis itself has prophase, metaphase, anaphase, telophase, and cytokinesis. So cytokinesis can be considered as a mitosis, or it can just be considered part of telophase. But anyways, so these different phases are really important. You'll go more into detail if you take the class. But anyways, then there's regulation of the cell cycle. So through this whole cycle, there are little checkpoints where things stop to make sure it can all catch up. For example, if there's a checkpoint after DNA synthesis to make sure that all your DNA has been replicated and it's all good to go. This way, it can prevent any sort of problems with replication. For example, you didn't have enough DNA to replicate, or your DNA is wrong, and then it produces the wrong cell. It doesn't have any of that happen. In cancer cells, though, this regulation usually isn't happening, so it'll continue to duplicate without having any sort of restriction, which is why cancer forms. Anyways, so the whole point of mitosis is to produce a cell that is exactly the same as the parent cell, or the cell that it was based off of. Okay, then we move on to heredity, where we learn about meiosis. So as opposed to mitosis, meiosis produces cells, daughter cells, that are different from the parent. So meiosis has two portions, meiosis 1 and meiosis 2. So all the phases are the same, like named the same. So there's prophase, metaphase, anaphase, and telophase, except meiosis happens like there are 2 rounds of it. So there's the replication of your DNA before meiosis 1, but not before meiosis 2. So in the end, in meiosis, you end up with half the number of chromosomes your parent cell has. This is because meiosis is to produce like babies and stuff. So when it starts out, it has both the mother and the father's DNA, and then it goes through the process, and then it ends up with just 1 pair, because a human only needs 1 pair and not 2. So the big difference between meiosis 1 and meiosis 2 is that in anaphase, here you can see there are 2 chromosomes and they're being pulled apart. So each chromosome is being pulled, so 2 are each being pulled apart. While in anaphase 2, you can see that each chromosome is being pulled into sister chromatids instead. So that's the big difference between the 2 meioses. So meiosis is able to produce genetic diversity, which is why humans are different from each other. We all have different genetic codes, and this is a big portion of why we are different. So the first thing is crossing over, or recombination. So during metaphase 1, these 2 chromosomes line up next to each other, and then they can exchange DNA, they can exchange information. So here you can see that the DNA from this blue one is exchanging with this red one, and in the end, it produces DNA chromosomes that aren't exactly as they started. So then there's independent assortment, which is and then also for crossing over, this can happen randomly. It doesn't, like, it can happen wherever on this chromosome, wherever it is just random. So that's what causes a lot of the randomness in your genetics. Then there's independent assortment, which is basically these during metaphase 1, these chromosomes can line up differently. Like it can be this or this. Well, see, this is only considering that if there only were 2 chromosomes in each cell. But anyways, they line up, and then they are able to line up differently, and then produce different organizations, like different ways that they are able to pair up, and this produces more genetic diversity. Okay, then we move on to genetics. So Mendelian genetics are the ones that you learn first because they're like the simplest form of genetics. And then so in Mendelian genetics, there are two laws. So there's a law of segregation and the law of independent assortment. Segregation means only one allele codes for each trait, while independent assortment means their traits are not linked at all. That means like if I have black hair and brown eyes, these are all independent. So it's the same chance of having black hair and brown eyes as like black hair and blue eyes, but that's not true because if you notice how most people who have brown eyes do have black hair, or black hair do have brown eyes, but it's because these two traits are linked, which I'll talk about in a second. Anyways, Mendelian genetics operates with simple dominance, meaning if there is a dominant trait, for example, if you look at this Punnett square up here, this big G is a dominant trait. So any plant with this dominant trait will have green, while even if it does have a recessive like a yellow, it'll still be green. So dominance is completely dominant. So Punnett squares are a tool used in genetics to simply be able to find some the ratio of offspring depending on the parents' genetics. So a lot of people don't like using Punnett squares, but they're really just a tool to help you be able to figure out these ratios. And then there's besides Mendelian genetics, there's also non-Mendelian genetics, which deals with stuff like codominance and incomplete dominance, as well as these other things. So in codominance and incomplete dominance, the Punnett squares and things are the exact same as in simple dominance, except for like in codominance, let's look at these flowers here. So in simple dominance, if red was dominant and white was recessive, in the simple dominance one, if there was a red allele and a white allele together, the flower would be red. So the heterozygous would be red. While in codominance, the whole thing, instead the flower would be pink. And in incomplete dominance, there would be patches of both. Then there's linked traits. So like I was talking before, the black hair, brown eyes thing, those are linked. This means if you think about it, I don't have a picture here, but on chromosomes, when they cross over, actually I'll go back to the past slide. When they cross over, if two traits are really close together, for example, they're like here and here, if they're really close together, then they're more easily to be crossed over together. So like they're here and then they cross over together to be here. But if they were far away, like here and here, it's less of a chance that they're going to cross over together, which is how they're linked. Then there's stuff like sex-linked traits, which are we learn, like you can also know that as X-linked traits because they're always passed down the X chromosome. So in this case, the trait again, it's on the X chromosome, which means that, for example, if you think about color blindness is a sex-linked trait. So it's sex-linked recessive, which means if you have two chromosomes and only the dominant one, which is non-color blindness, will show. Okay, so basically, in this case, for example, if a mother is a carrier, but the father does not have color blindness, when it passes down, you have boys have an XY while girls have an XX gene, chromosomes. And then it's different for females and males. So the way it passes down is slightly different from the way that a normal non-sex-linked trait would pass down. So on the AP exam, sometimes you're required to figure out, like, it'll give you some pedigree, like this one down here. It'll give you this and be like, what kind of how is this, like, is it sex linked? Is it just normal dominance? Stuff like that. It'll ask you for that kind of things. Is it recessive? Is it dominant? So it'll ask you what kind of trait it is without giving you any extra information. Then there's the last one of non-Mendelian genetics, which is having multiple genes control the expression of a trait. So for example, in hair color, different genes can make your hair this specific color it is. So just testing on one gene may not be able to determine anything. Okay, moving on. So then there's environmental effects on phenotype. For example, the rabbits, the snowshoe hares at the top. Snowshoe hares turn brown in the summer because of environment. It helps them blend in, and then it also helps them blend in to turn white in the winter because of the snow around, which is an environmental effect. Then there's chromosomal inheritance, which is basically all of the things like we talked about before. Okay, moving on. Gene expression and regulation. So in this unit, the most basic part is that DNA and RNA structure. So knowing that DNA is a double helix with 2 strands, while RNA is a single, and DNA has adenine, cytosine, guanine, and thymine, and RNA has adenine, cytosine, guanine, and uracil instead. So prokaryotes typically have circular chromosomes, while eukaryotes usually have linear chromosomes. So DNA replication happens in a lot of important places. For example, before mitosis and meiosis, there's DNA replication, and this happens it's like this. So DNA replication is semi-conservative, meaning one strand of the DNA is new while one strand is old. So like this blue strand is the original and the red strand is new. So half of it is new, half of it is old. And this is synthesized from 5' to 3', which means it can only be synthesized one direction. This is why things like lagging and leading strand happen. So because this DNA polymerase, which is the enzyme here, or the protein here, it can only synthesize this way. And this one can only synthesize this way because it's 5 to 3. But as you see, the split is here. So it's splitting here. So this on the lacking strand needs to move to the right. It needs to be moving this way, but it can only build this way, which is why it has to build, and then it has to jump over here and continue building and then continue like that, which is why there's a lagging strand and leading strand. Leading strand, it's synthesizing the same way that it's opening, which allows it to go faster and not need to jump around. Oh, oops. Okay, so next there's transcription, which is from DNA making into RNA. So DNA is transcribed into RNA similarly to how it's replicated, but not exactly the same. So it's just there's a template strand of DNA, and then the RNA is just built to match it. So after the RNA has been transcribed, it has to be processed. So during this processing, a 5' cap is added, a 3' cap is added, and then splicing occurs, which is just that introns are removed. So introns are just unnecessary part of the RNA that might not be, but then that won't be needed during the next step. So the splicing happens, and then the transcription, which happened inside the nucleus, the mRNA, the RNA which was made inside the nucleus, will now travel outside the nucleus to be translated into a protein. So, like I said, translation happens outside this nucleus at a ribosome, and it's where RNA is made into a protein or a polypeptide chain. So each group of 3 nucleotides is known as a codon, and it codes for one single amino acid. So in this case, the RNA is red, and then each one tRNA brings an amino acid and adds it to the growing chain of them. So here on the bottom right, you can see a picture of pre-RNA and then mRNA. So like this is what happens in the processing. For example, here like the light pink parts are unneeded, so they're introns, and then they get cut out through splicing, and in the end, this is able to travel through the nucleus and then get to the ribosome. Okay, moving on, we got regulation of the gene expression. So all of our cells in our body have the same DNA, the same amount of DNA, and it's all the same, theoretically. But our cells are specialized. So like our skin cells have the same DNA as like our hair cells, but these are expressed all the same. These cells are different because of the different expression of them. So operons are in prokaryotes only, like people know, usually think of bacteria. So for example, operons for like lactose, lactate, people being able to digest lactose in milk is based on an operon. Then there is gene expression and cell specialization. Like I said, the being able to have specific gene expression and not express all of our DNA allows for our cells to be specialized in what they do. Next is mutations, which are just the primary source of genetic variation. It's where there's like if there are different types of mutations, for example, point mutations or frame shift mutations, that basically mean a piece of genetic code, one nucleotide messed up, and then there's some sort of change to the expression of it. And then there's biotechnology, which is just the way we're able to work with this DNA. For example, gel electrophoresis, which is basically running DNA through an electric current and allowing it to flow through gel. And then PCR, which is polymerase chain reaction, which allows DNA to be multiplied and made bigger. So another big lab that we did was the pGlow lab. So basically it's where bacteria, E. coli specifically, is put in an environment with plasmids, which are tiny loops of DNA. So it has to uptake this DNA, and then we put it in plates full, some with nothing, and then some with like antibiotics, and see how it reacts. So we just learned how bacterial transformation, which is how bacteria takes up DNA from its environment, works, and then learn about this specific operon, which is why this thing will glow. So here's a picture from the lab that we did. Okay, next is natural selection. So natural selection was introduced by Charles Darwin, and it's a major part of evolution. So it basically states that evolutionary fitness is measured by reproductive success. So being able to reproduce is what defines how fit you are. So natural selection has a couple basic things, which is that environments apply some sort of pressure to a population, and then certain traits can increase or decrease the fitness of an organism in a specific environment. For example, if you have black mice, putting them in an environment with white sand, so like in a desert, will put a pressure, like if there was a pressure of having predators, they would be easily be able to see these black mice and this would decrease the fitness. This black fur would decrease the fitness of this mouse in the desert. But if this mouse was put in a place like on like rocks that are black, then this black fur can increase the fitness because it allows it to blend in and out of the eyes of predators. Artificial selection is just basically humans doing all this natural stuff, like humans, basically humans playing God, stuff like that. So then there's population genetics, which is like mutations, genetic drift, and migration. So genetic drift is basically how a big event or some sort of event can change the population of somewhere. So for example, the bottleneck effect, you can see like different like genes, different types with like with blue, white, and yellow, but some sort of bottleneck thing, like some sort of big thing happens so that only a couple survive, and in the end, your surviving population is different from your original. For example, here you can see the yellow, there are yellows in here, but there aren't any yellows in here, and after this reproduces, there probably won't be any yellows again. Then another large part of Unit 7 is the Hardy-Weinberg equilibrium. So this is just a math model to calculate changes in allele frequency, providing evidence for the occurrence of evolution in a population. So in short terms, it's just a bunch of math equations to allow you to predict how alleles will change. So there are 5 big conditions, which are: this must be tested on a large population, there can't be any sort of migration, there can't be mutations, and there can't be random mating. I mean, there has to be random mating, and there can't be natural selection. So like the mutation thing is already makes this like impossible to actually apply in the real world. But furthermore, like if the mutation thing was not considered, it still can't. It's very hard to find anything that will be really important, because usually all the important traits are either naturally selected for, or they're selected for during mating, which means the mating is not random. Here are the equations down at the bottom. I'm pretty sure these would be tested on the exam for like a lot of math questions. Okay, moving on. And then there's evidence of evolution, common ancestry, continuing evolution, and phylogeny. So evidence of evolution is basically like we have common ancestors. We all living things share basically the same type of DNA, or with the same type of coding and the different codons and the same amino acids. Common ancestry, basically we all share like common ancestors back to like some specific like bacteria, or like something closer would be like some people believe we share ancestry with like apes and primates. So also continuing evolution. So right now we're still continuing to evolve. The world is still continuing to evolve, and evolution is not only in the past. And then phylogeny is just like these phylogenetic trees over here. This is the most important part, is that knowing how to read them. These are able to show how recent a common ancestor was, how related we are to, or any organisms are to each other over the time. Then there's speciation, which is how one species is able to change into different ones due to natural selection. Extinction, which is when animals go extinct. And then variations in population, which allow, so variations allow species to withstand environmental pressures. For example, if we use that mice example again, if these mice are all on a white sanded desert, these black mice will stand out. But if they have like an offspring that is white because of some mutation, then it allows their species to continue to survive on, because this mouse is now more blended in better and then able to transfer its genes down to its offspring and allow the species to continue living. And then there's origin of life on Earth, which is like some history stuff about like how life started, the bacteria stuff. And then the final unit is ecology. So I don't have many slides on this, but ecology is basically how organisms and their environment interact to form an ecosystem. So organisms can respond to internal or external changes and through behavioral and physical mechanisms. So basically, and they can also communicate certain information to others, which is very important. Then there's like energy flow through ecosystems. So energy goes from producers to herbivores, and like you learn about the food webs in this. There's also population ecology, effective density of populations, community ecology, biodiversity, which is how like diverse an area is in like the types of animals or the types of organisms there. And then disruptions to ecosystems, which are usually like big natural disasters or like humans, which like with pollution or stuff like that. So now we're moving on to the free response portion, which is a deep dive into how this free response works. So going on a review from my very first presentation, this exam has two parts, a multiple choice and a free response, and I'll be going through the free response in depth. So you have 6 questions with 90 minutes, and it's 50% of your final grade. So your free response contains 6 questions, again, 90 minutes to complete, 2 long questions, and 4 short ones. So the long one is basically just a longer question with like more scenario, while the short ones are shorter. Okay, so College Board has their thing organized very well so that you basically can know what will be tested on every single question. This can all be found in the course exam and description, and you can just look it up. But all these things are shown, and you can all see what is going to be tested. So there's concept explanation, visual representation, questions and methods, representing and describing data, statistical tests and data analysis, and then argumentation. And then here, like which questions have which thing. So concept explanation is basically vocab. You have to be able to know what this specific thing is and be able to explain it out or write out what it is. Visual representation is basically usually it's stuff like charts, like any sort of pedigrees or like genetic charts or things like that. So you have to be able to explain the relationships with only these visual representation and be able to relate it like to a larger concept. Then there's questions and methods, which are basically anything to do with an experiment, like asking for hypotheses or predicting the results, identifying specific things, or finding like the variables or something. The fourth content is that there is representing and describing data, which is basically being able to find trends in like a table that they give you or describe relationships between variables. Then there's 5 and 6, which is 5 is statistical test and data analysis, which is basically math, being able to look at like a chart or something and perform some sort of math to prove something else. And finally is argumentation, which is being able to, if you had some sort of prediction before in a previous part, then you have to be able to prove that provide reasoning and justify why you think this is right through the information you're given. Okay, in question 1, you have this question is 8 to 10 points, and you have a scenario with a data table or a graph. So it assesses the following, so just like describing and explaining biological concepts, identifying experimental design procedures, analyzing data, and making or justifying predictions. Question 2 is also 8 to 10 points. It also gives a scenario and has data in a table. So it assesses student ability to do the following. For this one, you are expected to be able to construct a graph. So I feel like a lot of people don't know this, but like before I took biology, I didn't know how to do this either. But like you have to know how to plot error bars. So like on some, it'll be like plus minus some number, and then you have to be able to plot that. So if you don't know what that is, you could look it up and learn how to do that. Very important, because if you don't plot your error bars, you don't get your points. Okay. And then there's scientific investigation. So this is only 4 points. So the first 2 questions are a lot of points, while the last 4 are not as many. So you have to be able to describe biological concepts, all of that. It gives you a lab investigation instead of like a scenario. So keep that in mind. Question 4 is also a lab investigation, and being able to assesses the ability to do the following. And then finally, not finally, question 5 is again 4 points, giving you a lab investigation and being able to explain relationships and all the stuff listed below. And then finally, question 6, which is analyzing data, and it gives you a lab description and assesses the following.